Hi everybody. Today we shall discuss some of the questions from plant growth and development that can be asked in NEET. Usually 2 to 3 questions will be asked from this particular chapter. Let us discuss the important questions from this chapter one after the other. The first one. Plant growth is determinate with respect to the growth of. So usually root and stem they show indeterminate growth. Leaf, flowers, fruits, seeds, etc. They show determinate growth. Growth is only limited. So determinate growth is shown by leaf, taproot, stem, lateral roots. Taproot, stem and lateral roots they show indeterminate growth because of the presence of apical meristem. Whereas the leaf shows definite growth or determinate growth. So leaf must be the answer. Let us see the answer. Leaf. Here is the solution. Growth of leaf is determinate as they grow only to a limited extent. Growth of taproot, lateral roots and stem is generally indeterminate as they have apical meristem. Coming to the next question. A graph related to growth against time is plotted for a living organism growing in natural environment. It appears in the form of something related to the geometric growth. Right? Where graph is plotted, then that graph appears as sigmoid curve or S curve, not C curve, V curve, S curve, J curve. The information is given in the book itself. That is S curve. Let us see the answer. What one is the answer? Graph plotted growth against time appears in the form of sigmoid or S curve. Coming to the next question. Statement 1, Statement 2. Brachial elements are found due to differentiation and redifferentiation. Absolutely true. So, they are formed from the derivatives of procambium by differentiation. They are also formed from derivatives of lateral meristem or vascular cambium by redifferentiation. So, differentiation is transformation of derivatives of primary meristem into permanent cells is known as differentiation. Redifferentiation is differentiation of derivatives of lateral or secondary meristems into permanent cells is known redifferentiation. So, primary xylem is formed due to differentiation, secondary xylem is formed due to redifferentiation. And the tracheal elements are tracheids and vessels. Tracheal elements can show dedifferentiation, never. Tracheal elements are dead cells or dead structures. So, dead structures cannot show dedifferentiation. Dedifferentiation is regaining meristematic activity by permanent living and nucleated cells. So, statement 2 is wrong, statement 1 is correct and statement 2 is incorrect. First option must be the correct one. Let us see the answer. So, here it is the solution. Tracheal elements of primary xylem are formed from derivatives of procambium of apical meristem by differentiation. Tracheal elements of secondary xylem are formed from vascular cambium by redifferentiation. Since tracheal elements are dead, they cannot redifferentiate to form secondary meristems. Coming to the next question. At the cellular level, which are the common stages for development to both meristematic cell and cell no more in cell cycle? Common stages for development to both meristematic cell, dividing cell, and cell no more in cell cycle means withdrew from cell cycle or withdrawn from cell cycle, made exit from cell cycle. What are the common stages? Cell elongation expansion shown by both derivatives during differentiation as well as cell that retain the dividing ability of the initial cell. Plasmatic growth shown by both. Increased vacuolation shown by only the cells which are undergoing differentiation, but not by cells which have got the dividing ability. DNA replication, cells which have got the dividing ability only can show DNA replication, but not by the cells which are undergoing differentiation. So A and B are correct. Second option must be the correct one. Let us see the answer. 2. A meristematic cell and a cell withdrawn from cell cycle commonly show plasmatic growth due to RNA and protein synthesis and cell expansion or elongation. Increased vacuolation is shown by cells withdrawn from cell cycle and show differentiation. Reunification is shown by meristematic cells to prepare for next division. Next question. Assertion, reason type. Cotton shows heterophily. That is assertion. Yes, it shows heterophily. Here, heterophily is an example for plasticity influenced by stages in life cycle of a plant. Different stages in the life cycle of a plant. So, it's a case of developmental heterophily. Heterophily in cotton is influenced by environment. No, it is not influenced by environment. It is influenced by the stages in the life cycle of cotton plant. In that cotton plant, in the early stages of the development or seedling stage, it shows unlobed simple leaves. In later stages, it shows palmately lobed simple leaves. Assertion is correct, reason is wrong. A is correct and R is incorrect. Third option must be the correct one. Cotton shows heterophily. Juvenile cotton plant has got unlobed simple leaves, seedling stage. Mature cotton plant forms palmately lobed simple leaves. In this plant, the phases of life influence plasticity to form morphologically different types of leaves. Coming to the next question. This is a matching. So here in list 1, the chemical composition or chemical nature of the phytohormone or plant growth regulator is given. In list 2, the type of plant growth regulator is given. Indole compounds, auxins are indole compounds, A4. A must be 4. Adenine derivatives, cytokinins are adenine derivatives. So B3, A4, B3. C, carotenoid derivatives, abscisic acid derivative of carotenoids. Right? Carotenoids like xanthine, etc. Terpenes, gibberellins are terpenes. Gibberellins are diterpenes. So D1. So A4, B3, C2, D1. So answer must be the second option. Let us see the answer. Indole compounds, auxins. Right? Adenine derivatives, cytokinins. Carotenoid derivatives, abscisic acid. Terpenes, gibberellins. As I said, second option is given. That is the correct one. Coming to the next question. Auxins are growth promoters. So what you have to identify the correct statement. Auxins are growth promoters. Absolutely true. Auxins inhibit the growth of lateral or auxiliary buds during apical dominance. Absolutely true. Gibberellins were first discovered in rice plants infected by a fungus. Absolutely true. So they were first discovered in rice plants infected by a fungus known as Gibberella vesiculae. It is an ascomycetes fungus. Callus is a mass of undifferentiated cells. Absolutely true. During tissue culture, callus is formed. It is a mass of undifferentiated cells. Auxins and cytokines together play a role in differentiation. Auxins and cytokine together play a role in differentiation. Yes, in the regaining meristematic activity by permanent living cells, auxins and cytokines play a role. So we know that auxins also play a role in cell division. Cytokines also play a role in cell division. Together, they help in entering the permanent living cells into meristematic activity, that which is known as differentiation. So all the 5 statements are correct. A, B, C, D, and E are correct. Let us see the option. This is the correct one, fourth one. Auxins mainly promote growth. Auxins cause apical dominance and inhibit the growth of lateral buds. Auxin concentration inhibits the growth of lateral buds. Gibberellins were first discovered in rice plants infected by a fungus, Gibberella vesiculae. A mass of undifferentiated cells formed during tissue culture is called callus. Callus formation is promoted by a synergistic effect of auxins and cytokinins. Together, They help in the formation of callus. Callus is a mass of undifferentiated cell. Coming to the next question. Choose the statement not related to discovery of plant growth regulators. Cousins discovered the release of a volatile substance from ripened oranges. Related to the discovery of Italy. Correct one only. Abscissa 2 is a growth inhibitor. Absolutely true. Abscissa 2, dormin inhibitor B. So these are growth inhibitors related to abscisic acid. Miller coined the term kinetin. Yes, Miller only coined the term kinetin for the cell division inducing substance obtained from autoclaved DNA of living sperm. Crossover isolated auxins from oat seedlings. No. Crossover isolated, not isolated auxins. Crossover isolated gibberellins from the culture filtrates of Gibberella physicurae. He cultured the Gibberella physicurae and from the filtrate he obtained gibberellins but not auxins. So fourth one is not at all related. Crossover is related to gibberellins but not auxins. So fourth option must be the incorrect one, not related to discovery of plant growth regulators. Let us see. Fourth one is given. Crossover induced Botany disease symptoms by applying sterile culture filtrates of Gibberella physicurae on healthy rice seedlings. Thousands discovered the release of a volatile substance from ripened oranges that ripened raw bananas or unripened bananas. Abscissa 2 is a growth inhibitor, ABA. Miller coined the term kinetin for the substance obtained from autoclaved DNA that promoted cytokinesis. Coming to the next question. Choose the one not related to auxins, should not be related to auxins. Auxins are widely used in plant propagation through stem cuttings. Absolutely true. Because auxins promote the formation of adventitious roots in stem cuttings and play a role in vegetative propagation of useful plants. Auxins prevent early drop of roots and leaves. It's also true. They prevent abscission, early abscission of fruits and leaves. Auxins promote the drop of older, mature leaves and fruits. Yes, they promote the abscission of older one and mature leaves and fruits. First three statements are related to auxins. Auxins were first isolated from coleoptiles or canary grass. No. Auxins were first isolated from human urine. Right? So Bent isolated auxins from coleoptiles of oat seedlings. So fourth option must be the correct one. Not related to auxin. That must be the correct one. Fourth one is given. Auxins were first isolated from human urine but not from canary grass coleoptiles. Auxins promote adventitious root formation in stem cuttings in plant propagation. Auxins prevent early fruit and leaf drop but promote drop of old and mature fruits and leaves. Coming to the next question. Which is the most intensively studied gibberellin? It is gibberellic acid, which is nothing but GA3. It is the most intensively studied gibberellin. Not GA1, GA2, GA7. Third option must be the correct one. Let us see the answer. See? GA3 is the most extensively studied gibberellin. Coming to the next question. Statements 1 and 2. Gibberellins delay senescence. Yes, they delay the senescence of fruits like apple, oranges, etc. Gibberellins speed up the malting process in brewing industry. Absolutely true. Gibberellins promote the synthesis of alpha amylase, and this alpha amylase converts the starch into maltose. Right? So that's why second statement is also correct. First statement is correct, second statement is correct. Both statement 1 and statement 2 are correct. Fourth option must be the correct one. Fourth one is given. Gibberellins delay senescence of fruits. Gibberellins speed up malting process in brewing industry by promoting the synthesis of alpha amylase, which hydrolyzes the starch into a disaccharide maltose. Coming to the next one. Assertion reason. Yield of sugarcane increases upon application of gibberellins. Absolutely true, because gibberellins not only increase the length of the internodes of sugarcane, thereby the length of the stem is increased, but also they increase the sucrose content of the canes, thereby productivity increases. Sucrose content also increases, sugar content also increases. Assertion is correct, reason is also correct. Due to increase in the length of the canes, increase in sugarcane content, the yield of the sugarcane increases. Assertion is correct, reason is correct. Reason clearly explains the assertion. Here the option 4 must be given. 4. Gibberellins activate intracranial meristem of internodes of sugarcane and increase its length. They also promote formation of more sugar in the canes. This influence enhances sugarcane production. Coming to the next question. Which one is an influence of cytokinin on plants? Or cytokinins on plants? You have to identify the influence of cytokinins on plants. Promotion of apical dominance. No, cytokines decrease apical dominance. Promotion of flowering in pineapple. In pineapple, flowering is promoted by auxins and ethylene, but not by cytokinins. Bolting in rosette plants. Bolting in rosette plants is promoted by gibberellins, but not by cytokines. So naturally, the fourth option must be the correct one. Adventitious shoot formation, yes. So cytokines promote the formation of adventitious roots, contributes for stem growth, contributes for the formation of leaves, contributes for the green coloration of the leaves or chlorophyll synthesis. That also delay the senescence, cytokinins. Fourth option must be the correct one. Fourth one is given. Apical dominance is promoted by auxins, not by cytokinins. Flowering in pineapple is promoted by both auxins and ethylene. Gibberellins promote bolting in rosette plants prior to flowering, bolting in rosette plants like cabbage, cauliflower, etc. Cytokines promote leaf and chloroplast formation, lateral shoot growth, adventitious shoot formation, mobilization of nutrients to delay leaf senescence. Cytokines also decrease apical dominance. Coming to the next question, the matching. So, list 1, it is given the type of influence. List 2, type of growth regulator. You have to match them. Early seed production conifers, gibberellins. Right? Gibberellins promote the early maturity of conifers so that early seed production is promoted in conifers by the application of gibberellins. Xylem differentiation promoted by auxins. Very clear it is given in the book. Xylem differentiation is promoted by auxins. B1, A4, B1. Prevents apical dominance. Cytokinins are geatin. Nothing but a natural cytokinin. It prevents apical dominance or decreases apical dominance. D2. A4, B1, C2, D. Apical hook formation dicot seedlings by ethylene. D3. So A4, B1, C2, D3. So first option must be the correct one. Let us see the answer. Answer 1. Auxins promote xylem differentiation. Gibberellins promote early seed production in conifers. Geatin, prevention of apical dominance, right, and promotion of lateral buds or axillary buds into branches. Ethylene, apical hook formation in dicot seedlings. Coming to the next question. Which hormone is responsible for respiratory climactic? Is ethylene. Respiratory climactic is a sudden increase in respiratory rate during fruit ripening is known as respiratory climactic, promoted by ethylene. That is due to fruit ripening. Fruit ripening hormone is ethylene. Ethylene must be the correct one. Not abscisic acid, not cytokinin, not auxin. The second option must be the correct one. Let us see the option. Second one is given. Ethylene enhances the respiratory rate during fruit ripening, and the phenomenon is called a respiratory climactic. Abscisic acid promotes the senescence, dormancy, seed development, and seed maturation. Cytokines delay senescence of leaves. Auxins promote abscission of mature leaves and fruits and prevent the abscission of young leaves and fruits. Tenth one. Identify the correct statements with respect to physiological effects of ethylene on plants. You have to identify the correct statement. A, B, C, D, E. So such questions take a little more time to answer. That's why you should be very careful while reading the statements. Seed and bud dormancy is promoted by ethylene. No, it is broken by ethylene. Seed and bud dormancy is promoted by abscisic acid. Ethylene promotes sprouting of potato tubers. Absolutely true. It promotes the germination of seeds and sprouting of potato tubers. B is correct. A is wrong. A is incorrect. B is correct. Rapid internodal elongation of internode of deep water rice is promoted by ethylene. Absolutely true. So, in deep water rice during flooding, rice plants may be drowned. So, in such situations, ethylene is synthesized and it promotes the elongation of internodes so that the leaves are brought over the surface of the water. The leaves instantly they become aerial. So C is also correct. Ethylene enhances absorption of flowers. Absolutely true. Also enhances the absorption of flowers. Also enhances the absorption of fruits. So absorption is mainly promoted by ethylene, not by abscisic acid. D is also correct. Ethylene promotes the formation of more female flowers in monoecious plants like cucumber. Absolutely true. So ethylene promotes flowering in pineapple, synchronous fruit setting in pineapple. It also promotes the formation of more female flowers in monoecious plants like cucumber. What do you mean monoecious plants? Those plants in which male and female flowers are present on the same plant. Monoecious. The cucurbits are monoecious. When more female flowers are found, productivity also increases. So A is incorrect. B, C, D, E are correct. B, C, D and E are correct. Third option must be the correct one. Let us see the answer. Answer 3. Solution. Seed and bud dormancy is broken by ethylene. It promotes potato sprouting. In deep water rice due to flooding, ethylene promotes rapid internodal elongation to bring the leaves above the surface of water. Ethylene promotes absorption of flowers and fruits and helps in thinning. It promotes the formation of more female flowers in monoecious plant. It means ethylene, like cucumber, and enhances the productivity of cucurbits. Absorption of flowers and fruits and thinning in walnut. We have given example in the book. The other has given walnut, cherries, and cotton. For those crops, ethylene is sprayed for thinning or decreasing the number of flowers and fruits so that the quality of the fruit will be increased. Coming to the next question. Root hair formation is promoted by lateral root growth. Root hair formation is promoted by ethylene. So that the ethylene increases the absorbing surface area of the plant. So ethylene must be the correct one. Third option is given. Ethylene promotes the formation of root hairs and thereby increases the area of absorption of roots. It also promotes the formation of more lateral roots. Auxins promote the formation of adventitious roots. Cytokines promote the formation of adventitious roots. Adventitious shoots. Auxins promote the formation of adventitious roots. Cytokines promote the formation of adventitious shoots. Abscisic acid protects the plant from water stress. Next question. Which plant growth regulator ABA is antagonist in most of the situations? Given in the book clearly, ABA is antagonist in most of the situations to gibberellins. Not cytokinins, not auxins, not ethylene. So only little cases, cytokinins, auxins, ethylene, it is antagonist. But gibberellins mainly it is showing opposite effect. Answer must be the 2. Let us see the answer 2. ABA is antagonistic to most of the influences promoted by gibberellins. Coming to the next question. Choose the incorrect statement with respect to ABA influence on plants. Incorrect statement you have to choose with respect to influence of ABA on plants. ABA promotes seed development. True. Promotes seed maturation, seed development, etc. ABA promotes stomatal closure. Absolutely true. By promoting stomatal closure, helps the plant protecting from water stress. Hence, ABA is also known as stress hormone. ABA is inhibitor of plant metabolism. Yes. ABA is growth inhibitor and also inhibitor of metabolism. So 1, 2, 3 are correct statements. ABA was first discovered in connection with the fruit ripening. This is incorrect statement. ABA was discovered in connection with abscission and senescence dormancy, but not fruit ripening. Ethylene was discovered in connection with the fruit ripening. So fourth option must be the incorrect statement. Let us see. Fourth one is given. ABA was discovered in connection with its role in regulating abscission and dormancy. Coming to the next question. 2,4-dichlorophenolic acid is a natural oxygen used to control dicot weeds. No, it is synthetic oxygen. Synthetic oxygen used to control dicot weeds. Yes, it is correct. Ethylene releasing substance used for thinning in cotton and cherry and walnut. No. Synthetic cytokinin used to prevent apical dominance in plants. No. Synthetic oxygen used to control dicot weeds. Second one is correct. 2,4-dichlorophenolic acid is synthetic oxygen used to eradicate dicot weeds. I request you to like, comment, subscribe, share, click the bell icon to get the notifications of my post. Thank you very much.